and welcome to Spotlight on Antibodies, a podcast from BioWintron, where we bring you the latest antibody industry trends. This month in July, we'll highlight antibodies for IVD applications, the druggable proteome, and degrader antibody conjugates. Antibodies are used as research tools, therapeutic drugs, and also for in vitro diagnostics, or IVDs. These are tests performed outside the body, using samples such as blood, urine, saliva, or swab extracts. Antibodies can be raw materials in many common IVD applications, including enzyme-linked immunosorbent assays, chemiluminescent immunoassays, lateral flow tests, and particle-based immunoassays. Demand for these materials is growing. One recent market report projected that the global IVD raw materials market would increase from approximately $27.5 billion in 2025 to more than $30 billion in 2026. But supplying an antibody for a diagnostic assay involves finding one that binds the intended target, identifying combinations that work together, confirming their performance for the intended assay format, and eventually establishing a production process that can provide sufficient material with consistent quality. This matters because variability in these raw materials can affect the sensitivity, specificity, and reproducibility in the final diagnostic system. One example is the sandwich immunoassay. As the name suggests, the target antigen is positioned between two antibodies. One antibody acts as the capture antibody, attaching the antigen to a solid surface. A second antibody acts as the detection antibody, producing a measurable signal. In principle, this sounds straightforward. In practice, the two antibodies must be carefully matched. They need to bind different accessible sites on the antigen without blocking each other. Their binding must also remain effective after one antibody has been immobilized on a surface and the other has been labeled or conjugated to a detection reagent. This means that an antibody with strong binding activity on its own may still perform poorly when paired with another antibody. Testing individual antibodies cannot replace testing the combinations themselves. During antibody pair screening, researchers compare different capture and detection arrangements. They assess whether the epitopes are compatible, whether both antibodies retain sufficient binding activity, and whether the pair distinguishes the intended target from related proteins or other components in the sample. They also examine the assay's detection range. The pair must generate a useful signal across the antigen concentrations that are clinically or analytically relevant. Researchers must also compare a positive signal with the background observed in negative samples. A pair that generates a strong signal but also produces a substantial nonspecific background may be less useful than a pair with slightly weaker signal but a much better signal-to-background ratio. In some cases, researchers may also need antibodies with different affinity ranges. This can help support measurements at both low and high antigen concentrations and potentially extend the assay's linear detection range. Modern antibody discovery platforms may generate hundreds or thousands of candidate sequences. But before researchers can select the best antibodies or antibody pairs, those sequences must be expressed and tested experimentally. Small-scale parallel production can provide enough material for initial binding studies, pair screening, and early assay evaluation. Antibody performance can also change depending on whether the assay uses a coated plate, a membrane, a particle, or another surface. Conjugation chemistry, incubation time, flow conditions, buffer composition, sample type, and detection method can all influence binding and background. Even the antibody's constant regions, the parts outside the variable region that directly recognizes the antigen, can influence expression, stability, immobilization, or interactions within the assay. As the assay moves from early screening into optimization, validation, and stability studies, antibody requirements increase from small experimental quantities to larger and more consistent batches. recombinant antibody production can support this transition. Once the heavy and light chain sequences are known, the antibody can be produced from a defined sequence. Recombinant systems can also produce different formats, including full-length antibodies, Fab fragments, and single-chain variable fragments. The appropriate format depends on the design and requirements of the diagnostic assay. BioIntron supports this progression from early screening to continued antibody supply. RushMab can produce small quantities of multiple recombinant antibody candidates for experimental testing and pair screening. Once a sequence and antibody pair have been selected, the project can move into larger-scale production for optimization, validation, pilot manufacturing, or ongoing kit production. For decades, many disease-driving proteins have been described as undruggable. Some lack the binding pockets needed for conventional small molecule drugs. Others are difficult for biologics to reach. Antibodies generally bind targets on the cell surface or in the extracellular environment. But many important cancer drivers, such as Cras and p53, operate inside cells. New antibody formats and engineering strategies are beginning to work around that limitation. They are finding alternative ways to recognize or act on intracellular biology, with one approach taking advantage of the cell's own protein processing machinery. Proteins inside cells are continuously broken down into short peptide fragments. Some of these fragments are displayed on the cell surface by the human leukocyte antigen, or HLA molecules, where they can normally be inspected by T cells. TCR-like antibodies, also called TCR-mimic antibodies, are designed to recognize these peptide HLA complexes. In effect, the antibody can detect evidence of an intracellular protein without having to enter the cell itself. Researchers recently applied the strategy to Cras G12D, a cancer-driving mutation found in several tumor types. A team combined computational antibody design with experimental screening to identify TCR-like antibodies, recognizing a Cras G12D-derived peptide presented by HLA. Their selected antibodies showed strong selectivity for the mutant complex over tested controls. However, peptide HLA targets introduced another problem as they can present at very low levels on the cell surface. This is where bispecific antibodies may help. One arm can recognize the mutation-derived peptide HLA complex, while the other binds CD3 on T cells. The goal is to recruit T cells directly to tumor cells displaying the target. This concept was demonstrated preclinically, but these therapies may depend on a patient's HLA type, how efficiently the peptide is processed and presented, and whether tumor cells maintain sufficient HLA expression. A different strategy uses the antibody not to recognize the intracellular protein, but to deliver something that can act inside the cell. Antibody-drug conjugates already use this principle with cytotoxic payloads. Researchers are now exploring broader payload classes, including oligonucleotides and molecules designed to alter or degrade intracellular proteins. Computational protein design adds another dimension. Rather than relying entirely on previously discovered binders, de novo approaches attempt to design antibodies against specified regions of a target. ABSCI, for example, uses the term zero prior epitopes for target sites without repeated antibody complex structures. In their recent work, their platform generated experimentally validated binders against several such target regions. This doesn't make intracellular proteins directly accessible, but illustrates how computational methods can expand the range of epitopes that antibody discovery can explore. Importantly, computational design doesn't remove the requirement for experiments, as these antibodies still have to be tested for binding, specificity, cross-reactivity, stability, aggregation, cellular activity, and manufacturability. Together, these technologies are gradually expanding what an antibody-based therapeutic can be designed to target, and what it can be designed to do. One of the clearest examples of how antibody technology is moving beyond conventional payloads is the emergence of degrader antibody conjugates, or DACs. Like an antibody-drug conjugate, a DAC uses an antibody to recognize a surface antigen and deliver a payload into selected cells. But instead of carrying a cytotoxic drug, the payload is a targeted protein degrader designed to eliminate a specific intracellular protein. That difference changes the mechanism. Targeted protein degradation instead uses the cell's own protein disposal machinery. Most degraders recruit an E3 ubiquitin ligase to a protein of interest, marking that protein for degradation by the proteasome. A DAC combines these two ideas. The antibody binds an antigen on the target cell and is internalized. After intracellular processing and linker cleavage, the degrader payload is released. It can then engage its intracellular target and recruit the machinery needed to degrade it. A free degrader may act wherever it distributes in the body. A DAC is designed to concentrate that activity in cells expressing a particular surface antigen. In principle, this could make it possible to degrade intracellular proteins that would be difficult to target systemically, because they are widely expressed, difficult to inhibit, or associated with toxicity in healthy tissues. However, internalization doesn't mean that active payload reaches the cytosol. The linker has to remain stable during circulation, but release the degrader efficiently inside the target cell. The payload itself has to retain activity after conjugation and processing, and the relevant E3 ligase has to be available in the right cellular context. Despite these challenges, commercial activity has increased. In July 2026, Johnson & Johnson completed its acquisition of Firefly Bio for $1 billion, gaining its DAC platform and preclinical programs focused in part on KRAS-driven cancers. Earlier in the year, Roche expanded its collaboration with C4 Therapeutics to develop DACs against 2 undisclosed oncology targets. And the field has started to reach the clinic. BMS 986497, originally developed by Aurum Therapeutics, is being evaluated in a Phase 1 study in acute myeloid leukemia. Aurum is also advancing a CD123 targeting GSPT1 degrader towards clinical development. DACs remain an early-stage modality, and important questions about safety, effective intracellular delivery, resistance, biomarkers, and therapeutic window still need to be answered. For preclinical researchers exploring antibody conjugates and new payload classes, conjugation and analytical characterization are therefore important parts of evaluating whether a construct behaves as intended. BioIntron's ADC conjugation platform supports antibody expression, conjugation, and quality assessment for preclinical screening and evaluation. And that's all for this month's antibody industry trends. To learn more insights into antibody drug news, make sure to follow BioIntron on LinkedIn or our website at www.biointron.com. Keep up to date with further episodes from Spotlight on Antibodies by following this podcast.